Hello everyone, and welcome to today's live broadcast, Developing Personalized Cancer Neoantigen Vaccines, presented by Agnete Fredriksen, Chief Scientific Officer, Vaccibody AS. I am Alexis Corrales of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Accreditation button located in the promotional board at the bottom center of your screen and follow the process to obtain your credits. Please join me in welcoming Dr. Fredriksen. I will now turn the presentation over to her.
And I'm happy to be here and allow you to hear about Vaccibody and what we are doing in the cancer neoantigen vaccine space today. So if we go to the agenda for today, I'll first give you some background on cancer vaccines and in particular on the neoantigens. And before I start telling you about Vaccibodies cancer vaccine strategy, and importantly in the neoantigen vaccine space, we also need to discuss neoantigen prediction tools. And then I will end up today talking about Vaccibodies clinical trial experience and our future plans. So we in Vaccibody, we're working within immunotherapy, which we believe is the next wave of cancer therapy. This is cancer care is based on these 5 pillars, and recently there's been a lot of success within immunotherapy. This is based mostly due to the success of the checkpoint inhibitors, but also vaccines and cell therapies and others falls within this category. And we believe that cancer vaccines are the best strategy suited to stimulate the control and then also truly specific individualized immune response. So the checkpoint inhibitors has revealed the importance of the immune system and its role in fighting cancer. So we have observed over the latest years that if you provide checkpoint inhibitors to late-stage cancer patients, we can increase the survival rate. And in particularly interesting, we can see very long-term survivors that's been treated with checkpoint inhibitors. So the checkpoint inhibitors and their mechanism of action is to allow the immune system to continue to work, to not inhibit the responses of the immune system, and in particular of T cells. What was also observed in the patients that had clinical benefit on checkpoint inhibitors was that these actually had a high neoantigen burden, and this is where we come into the word neoantigens. It's basically due to mutations in the cancer cells that are not present in the healthy tissue of that particular presence. And actually what was observed was the patients that had a lot of mutations and there's a high neoantigen burden had the higher possibility to respond to checkpoint inhibitors than the patients with less mutations and thus low neoantigen burden. This means that in particular, for instance, for lung cancer patients, the patients that had been exposed for heavily smoking had a higher chance of benefiting from checkpoint inhibitors because they had exposed their lung cells, cancer cells, for a lot of mutations. However, There are still modest survival rates by just providing checkpoint inhibitors to cancer patients. And looking at the mechanism of action, we know that the checkpoint inhibitor limits the responses to the already existing T-cell repertoire in the cancer patients. So it allows the T-cells that are already existing to fight tumors if they are specific to the tumor cells, but it will not necessarily increase the T-cell responses, and we will not expand T-cells that are not already present in the patient. It also revealed a very important role of these mutations of neoantigens as a very good target for the immune system in cancer patients, in cancer immunotherapy. So the question then is, how can we best boost the immune response to elicit more effective and broader neoantigen-specific T-cell responses? When we look at the different indications, cancer indications, there are generally different number of mutations in the cancer cells based on the different indications. So neoantigens, they are tumor-specific mutations, not tumor-associated self-antigens or self-antigens. So these are mutations that we only find in the cancer cells. And for instance, melanoma and lung cancer are the indications where we do find the highest number of mutations due to exposure of UV light and smoking, for instance. It gives a high mutational burden in those indications, and that also correlates with the indications that shows most promise of the checkpoint inhibitors on a general level. So the idea then with these neoantigens, there are newly arising mutations that happen in the T cells that could be really good targets for the immune system, because these are truly tumor specific, and we should not have tolerance developed to the new neoepitopes that's been formed by the mutations in the cancer cell. So if you look closer up the T-cell immunity to infectious diseases and to cancer, we do have a very high success of vaccines targeted to infectious diseases. So in the cases where this is T-cell dependent, there needs to be a T-cell receptor, an infected cell with an HLA molecule that is presenting the epitope from the virus or the bacteria to the T-cell receptor. So this is then, the antigen is then in this case foreign because it's of viral or bacterial origin, and it's recognized by a T-cell receptor that's not been tolerized. It's actually there patrolling the body to see if there is any foreign substances that is invading the body. However, in the cases where we have pursued the same principle with cancer self-antigens or tumor-associated antigens, we tried the same principle, but the HLA molecules presenting the self-antigens and tumor-associated antigens, they are not foreign to the immune system, and we have already developed tolerance. So the T-cell receptors with high specificity to the self-antigens, tumor-associated antigens, would have been deleted in the thymus, and thus the very strong specificity will not, we cannot really raise a strong T-cell response to self-antigens. And thus we have had numerous failures with cancer vaccines trying to increase the T-cell response to self-antigens. So with these neoantigens, so it's a self-antigen in principle, but it has at least one mutation from the wild type sequence to this then new mutated sequence. So in this case, we need to understand if the immune system is able to observe the difference when a self-antigen expresses slightly changed epitope and displays this to the T-cell receptor. And this, we now know that it can be observed as a very foreign epitope, and T-cell receptors with high specificity may be able to recognize these neoepitopes as a strong T-cell epitope, dependent on the mutation and dependent on a lot of factors on the origin of the self-antigen as such. So this is what we're pursuing in the cancer neoantigen vaccine field. We want to raise an immune response to these neoepitopes in our self-proteins so that the T cells, our own immune response, will recognize the cancer cells as foreign and attack the cancer cells. So we observed lately that neoantigens can really be strongly immunogenic tumor antigens. There is a study that was published in Nature in July by Sahin, where they first diagnosed patients with melanoma and started to manufacture neoantigens vaccines that were specific for each individual patient. That requires a lag time when it comes to manufacturing, because you really need to manufacture one vaccine per patient. In the waiting period, they picked shared antigens, tumor-associated antigens, and vaccinated the patients while they were waiting for the neoepitope vaccines. And you can clearly see from the results that the immune responses to the shared antigens were not as strong to the immune response that they were able to elicit to the neoepitopes. There are higher affinity T-cell receptors available for neoantigens in circulation, and the interferon gamma T-cell responses to the neoantigens can thus be stronger than to the shared tumor-associated antigens. So this is a fairly new concept. So there are two clinical studies with personalized neoantigen vaccines, both in melanoma patients, that were published in Nature in July, with 6 or 13 patients with close to 100 neoepitopes in both studies tested in these patients. These were either vaccinated with peptide vaccines or with RNA-based vaccines. But the results were quite promising. They did observe T-cell responses to between 60 and 76% of the neoepitopes that were selected for the patients, and with strong immunogenicity. However, it was mostly CD4 T cells that they were able to raise towards the neoantigens, and not that successful CD8 T cell responses. However, so these were patients in an adjuvant setting, so they did not have a high tumor burden. And it's a bit difficult to speculate about the clinical responses, although it does seem to be very promising so far. But we're still awaiting studies in cancer patients, late-stage cancer patients with a high tumor burden. I think what we observed in the study was that more than 50% of the neoepitopes were immunogenic. The cancer vaccine strategy is able to increase the immune response to de novo T cells that already were present, specific to neoantigens in the patients. That was observed in approximately 1/3 of the of the neoepitopes that were tested, but also raised de novo T-cell responses to neoantigens that could not be found in the patients before vaccination. So 2/3 of those were towards de novo neoepitopes. And also importantly, most of the responses observed were CD4 helper T-cell responses, and not the CD8 T-cell responses that we've learned classically that we want to raise in order to have a strong response towards the cancer cells. So there are a lot of promises to vaccinating patients with neoantigen-based cancer vaccines, because we now know that it might be very effective if we are able to raise T-cell responses to these newly mutated tumor-specific antigens. However, what we learned is that these tumor-specific mutations are practically all the time patient-specific. So it is very, very rarely that we can find the same mutation in 2 different patients, and also that mutation is a perfect match with their HLA molecules. So what we are actually pursuing is to make one vaccine specifically designed per patient. That means that we have to go through these processes where we first take a biopsy from the tumor, and we also harvest healthy tissue or blood samples. We sequence them both, and we compare so we can identify the tumor-specific mutation in that particular patient. And based on the mutations and its relationship to the wild type and different other features, we do a lot of bioinformatics in order to select the neoepitopes that can be promising in a vaccination setting. And then we have to move on to manufacture the vaccine under GMP, and that will be 1 vaccine per patient. And then the vaccine needs to be shipped back to the site, clinical site, administered, and it needs to be in a format that can elicit strong immunogenicity and hopefully provide clinical benefit. So in order to go through this entire process and specifically design one unique, perfectly fit vaccine per patient, it is essential that this can be done within a reasonable time, at reasonable cost, and with very strong efficacy. And that's what we are pursuing in Vaccibody. So then I can tell you about Vaccibody and the cancer vaccine strategy that we're using within this space. So Vaccibody is a biotech company. We have 1 clinical study where we just finalized enrollment in the phase 2A. This is called the VBC-01 study, and it's a monotherapy where we treat patients that have developed precancerous cervical lesions with a therapeutic HPV-16 cancer vaccine. And then we are now heading into a new study within the neoantigen vaccine field, where we will set up a basket study treating patients with melanoma, lung cancer, bladder, renal, head and neck. That will be initially 40 patients, and the aim is to have the first patient in within this quarter. So Vaccibody was originally developed based on the concept of targeting antigens to antigen-presenting cells in order to create more efficacious vaccines. So we know that every antigen needs to be processed and presented by antigen-presenting cells on HLA molecules to T cells in order to elicit a strong T cell response. And we make sure that this happens effectively by creating the vaccine in this specific format that is created as a DNA vaccine plasmid that is encoding 3 different modules that we call functional modules. One is the targeting unit that targets the antigen to antigen-presenting cells. One is the dimerization unit that cross-links the target receptors. And then we have the antigenic unit. So the vaccine is actually administered as a naked DNA plasmid and allows us to have in vivo expression of the protein format, which actually simplifies the manufacturing process dramatically. And that is very important when you want to create one vaccine per patient. So the mechanism of action, the DNA plasmid that encodes this Vaccibody fusion protein will be delivered intramuscularly. It will enter the muscle cells, and the muscle cells, the myocytes, will produce and secrete the protein format of the vaccine. So it will be in vivo protein expression and secretion. And then the targeting unit in our clinical products is a naturally occurring inflammatory chemokine. And this chemokine has the ability to target and attract antigen-presenting cells. Its natural function is to alert the immune system when there is an inflammation. So the antigen-presenting cells will actually travel to the injection site due to the high concentration of the Vaccibody protein with these chemokines locally. And then the targeting unit, the chemokine, will bind to the chemokine receptors on the surface of these antigen-presenting cells. It does so in a dimeric manner. And when it binds these 2 chemokine receptors, it will cross-link the receptors. And this provides an activation and maturation signal to the antigen-presenting cells. And then the entire complex is taken up by receptor-mediated internalization and broken down and presented on MHC class 1 and MHC class 2. Another interesting feature with this particular chemokine and its ligand, the chemokine receptor, is that they are described to be particularly good at entering this cross-presentation pathway and allowing effective presentation of the neoantigens than of the antigens on HLA class 1 and effectively stimulating CD8 positive T cells. So we know that the Vaccibody can target dendritic cells. We know here that it can attract the antigen-presenting cells. So we can actually, in the middle here, we can inject the Vaccibody encoding a construct encoding M-cherry. So we can observe that the transfected cells will here be shown as red, and they will be infiltrated by antigen-presenting cells. So this will happen locally, and we will have very strong delivery of the antigens to the antigen-presenting cells. We also can see that the antigen-presenting cells mature upon binding the Vaccibody protein and upregulate CD80 and CD86, for instance, shown in this slide. Another nice feature with the Vaccibody is this local attraction, due to the local attraction of the antigen-presenting cells. We can deliver our vaccines with this simple jet injector. It's a needle-free jet injector. It's very small, handy, easy to use, and very minimal pain. This is cost-effective and applicable for multiple immunizations. And the reason why I bring this up is that normally DNA vaccines has been shown to be less immunogenic, and also that in order to have an effective immune response to the antigens delivered with a non-targeted DNA vaccine, you need to deliver it in combination with in vivo electroporation, which is quite painful and requires electricity and quite costly devices. And it is can provide a lot of pain to the patients. So that's an added feature of this attraction of the antigen-presenting cells, and we've shown that with this targeting unit, we can achieve the same level of efficacy with this PharmaJet needle-free jet injector instead of using in vivo electroporation. So when it comes to the neoantigens with Vaccibody, we have gained a lot of expertise in preclinical studies, testing a different number of neoepitopes, different lengths of neoepitopes, different order and different linkers of the neoepitopes in order to find the optimal robust vaccine format that we can move into the clinic. So at this stage, we tested more than 70 different BB10 neoconstructs with more than 400 neoepitopes to date. And that gives us a lot of comfort moving into the clinic, and we found the optimal format. We will move into the clinic with the format where we can have between 10 and 20 different neoepitopes in one vaccine, and all neoepitopes will then be from the same cancer patient. So we'll have multiple targets in one patient, but the manufacturing is just one vaccine per patient. So we've tested the format, and we have a lot of experience with the format, knowing that it can provide both rapid, strong, and long-lasting immune responses compared to non-targeted vaccine delivery. With the neoepitopes, we use the information in the literature where neoepitopes have been already described for a few mouse tumor models, B16 in this case, and compared delivery of the exact same neoepitopes in the Vaccibody format, BB10-neo, head-to-head with giving the exact same peptide with the same sequence in combination with the poly-IC adjuvant with 2 different doses. And we could confirm that the immune responses to the neoepitopes were both stronger and broader with the Vaccibody vaccination delivery system than with peptides and adjuvants. Interestingly, we also observed that it was not necessarily the same neoepitope that dominated amongst the 10 neoepitopes if you give them as a peptide as if you give them in the Vaccibody format. So if you see that the epitope number 9, PEP-M9, the purple one is the strongest one if you give it as peptide. But with the Vaccibody, peptide number 10 is a very strong epitope, but this is not immunogenic at all if you give it as peptide. So recently, we've looked deeper into exactly the background for the differences in these results. So the immune response to the same 10 neoepitopes, if you give them with a peptide and poly-IC adjuvant or as an RNA vector, has been published before. And even though they were all originally picked based on bioinformatics that showed that these could be potentially strong epitopes binding to MHC class 1 and thus eliciting CD8 T-cell responses, most of the T-cell responses observed towards these 10 epitopes, if you give them as peptide or RNA, were CD4 T-cell responses. Also, the immune response profile, if you gave them as peptide or RNA, these are published in the same papers, seem to be quite identical, but not really fully overlapping. And then when you deliver exactly the same neoepitopes in the Vaccibody VB10 Neo format, we observed a completely different picture. So we did actually observe a very strong CD8 T cell response to more than 50% of the neoepitopes in this case. And we observed a CD4 T cell response to all 10 neoepitopes. So it was broader, but it was also more CD8 T cell responses in this case. And we, interestingly enough, to all the neoepitopes where we observed a CD8 T cell response, we could also see a CD4 T cell response. And it was quite different from the RNA and peptide published data. When we look in the next slide, it's the same data where you can actually really see the differences in the immune response. It's almost opposite. So if you look at peptide M2, M3, M4, M7, and M10, these are the ones where we observe a very strong CD8 T cell response combined with a CD4 T cell response. And those are the epitopes that are the weakest, either non-responding or very weakly responding if you give the exact same sequence as a peptide and poly-IC vaccine format. So it really shows that the vaccination system is extremely important to elicit the right T cell response to the exact same antigen. And then, even though you are able to predict a very strong CD8 T cell epitope, it cannot be immunogenic if you do not deliver it in the optimal vaccination format. Another thing we've looked into is the number of neoepitopes and the importance of having a technology where you can where you can insert multiple neoepitopes in one vaccine. So we tried a system where we had 10 neoepitopes and the other where we had 20 neoepitopes, shown in red and blue here. So we challenged mice with tumor cells, and then we measured the tumor growth. And we vaccinated mice then with the construct, Vaccibody constructs, either holding 10 neoepitopes or 20 neoepitopes. And we could really see that there was a huge benefit of using multiple neoepitopes, and the breadth of the immune response can be important. And we believe this is even more important moving into the clinic and taking into consideration the heterogeneity of the tumor and the different metastases. So it's also a huge advantage to have a technology that can hold multiple neoepitopes in one construct when you have to manufacture one vaccine per patient. In contrast to peptide vaccines, for instance, where each peptide is manufactured individually. Moving into the neoantigen prediction tools, I think the data that I just showed that it's a very different immune response to the exact same neoantigen, whether you deliver it as a peptide RNA or Vaccibody DNA vaccine, that led us into trying to develop our own neoepitope prediction tool based on data, in vivo data, where we use our own delivery vehicle in mice and looking into the immune response to each individual neoepitope and looking into then all the features of the neoepitopes that are strongly immunogenic, weakly immunogenic, and non-immunogenic, and training the selection tool based on the data that we achieve vaccinating with our own delivery, Vaccibody delivery device. We've done this now with close to 400 neoepitopes. And started to see very strong pattern. This is the pattern, this is the selection tool that we will be using moving into the clinic. We recently tested our Neoepitope prediction tool, the latest version, on a completely uncharacterized tumor model, the LL2 lung cancer tumor model. And in this case, we were able to find 68% immunogenic neoepitopes with a very strong total immune response. So we are very confident moving into the clinic with this prediction tool based on our own data. So we're then moving into the clinic. We now received a conditional approval to start our VBNO1 trial. So we plan to recruit the first patient here in Q1 2018. Importantly for Vaccibody, our previous program with the therapeutic HPV vaccine has given us a lot of benefit moving into the clinic with the neoantigen-based cancer vaccines. It's actually the exact same construct that we will be using. It's the DNA plasmid, same plasmid backbone, same targeting unit, the chemokine, same dimerization unit. The only difference is the antigenic unit, which in this study was 2 full-length proteins from HPV-16, while in the neoantigen studies, these will be neoepitopes that will be specific and different than for each individual patient. So actually, everything that each individual patient in the neoantigen trial will receive, those components will already have been tested in the patients in our HPV trial. So in this study, we learned that we did not observe any drug-related SAEs. So as a monotherapy, the Vaccibody vaccine is very safe. We tested actually 2 different dosing regimens in that study. So we learned the importance of having a quite tight vaccination interval. 3-week intervals induce the strongest immune response. So we will move that forward into the neoantigen study. And we did also observe very promising clinical efficacy that strongly correlated with the T-cell responses in this study. So we have information both on safety, efficacy, dose regimen, combination strategies, and manufacturing experience from this study that we take advantage from when we move into the next VBNO1 study. So this study is an open-label first human dose to evaluate safety, feasibility, and efficacy of multiple dosing with the VB10 Neo immunotherapy. It will be performed in patients with locally advanced or metastatic melanoma, non-small cell lung cancer, clear renal cell carcinoma, urothelial cancer, and squamous cell carcinoma of the head and neck. These are all indications that have approved checkpoint inhibitors as standard of care, and they all have a moderate to high mutational load. So in this study, in order to learn as much as possible of the contribution of the Vaccibody vaccine, but also take advantage of the synergistic responses of vaccination and checkpoint inhibitors, we will enroll patients that's been treated with checkpoint inhibitors for at least 12 weeks, and then start manufacturing the VB10 Neo vaccine. And in this case, we will enroll patients then that have not received a complete response on checkpoint inhibitors, and they're also not rapid progressors, but the intermediate responders. And then we will add vaccines, and there will be a few months between they started checkpoint inhibitor therapy and when they start the VB10 Neo vaccinations, which hopefully will allow us to dedicate the clinical responses as well, more likely to be due to the vaccine. than only the checkpoint inhibitors. We are working with, very happy to be working with these 3 renowned international clinical sites in Germany. Work with Jürgen Kraus in Heidelberg in Germany, Professor Angela Krakart in Munich, and Professor Elke Jäger in Frankfurt. They are all have a lot of experience with early immunotherapy trials, with checkpoint inhibitor therapy, and also have a special interest within neoantigens. So going back to this slide with the processes that we will go through for each patient, Vaccibody has then developed its own VB10 neo-specific proprietary selection method that we believe will be the key in order to find the most immunogenic neoepitopes compatible with our vaccination principle. And then again, importantly, what we observe is that this vaccination technology can increase the likelihood of engaging CD8 T-cell responses to the neoepitopes. For the manufacturing, we do deliver the VB10 neo vaccines as naked DNA plasmid in PBS. It's a very rapid, robust, cost-effective manufacturing. DNA plasmids are stable and safe, and the format allows us to hold up to 20 neoepitopes at least per vaccine, which means that the vaccine manufacturing process is cost effective. And then when it comes to the delivery, we have the needle-free delivery, patient-friendly, and the mechanism of action of the Vaccibodies provides a very rapid, strong, and long-lasting response. And we also see that it's broad and CD8 dominated. So we believe that we can fulfill the requirements of having a rapid, cost-effective, and efficacious vaccines moving into this new exciting field. So this is the core team in Vaccibody at the headquarters here in Oslo in Norway. Really looking forward into starting the clinical trial and see how this pans out in patients. So thank you for listening in, and then I'll take any questions from now.
Thank you, Dr. Fredriksen, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for, so let's get started. Our first question is, manufacturing of one vaccine per patient seems challenging, both from a logistical and cost perspective. Which steps do you consider as key factors to bring individualized neoantigen-based cancer vaccines to the market?
Yeah, so absolutely. This can be quite challenging, and bringing it to the market, I think it's extremely important that you have this technology that can hold a number of neoepitopes in the same vaccine, so you don't have to manufacture multiple vaccines per patient. It's also important to have a robust technology, and manufacturing DNA plasmids should allow this to happen also in a robust and rapid manufacturing technology. At this stage, I think there are multiple steps so that we can optimize. And one of the features you can do is to put everything in one dedicated facility. So you have all the steps from sequencing to neoepitope prediction and manufacturing and release under the same roof. And that is something that will happen in the future and will shorten the time period as well. But I think the key is to have a vaccine technology that allows a robust, rapid manufacturing as DNA plasmids.
Our next question is, DNA plasmids are stable and can be easily manufactured, but has shown limited success as vaccine delivery vehicle. How does the Vaccibody differ in mode of action from traditionally DNA plasmids?
Good question. I think it's multiple factors here. Importantly, a normal DNA plasmid does not have this targeting unit. It relies on direct transfection of antigen-presenting cells. So in that case, you want the DNA plasmid to transfect an antigen-presenting cell so that that antigen-presenting cells can present the antigen that is delivered directly on the surface on the MHC molecules. That does not necessarily happen as targeted as you want. You also need to activate the antigen-presenting cells. And what we do with the Vaccibody technology, we make sure that in principle all the antigens are delivered to antigen-presenting cells, not through the transfection of the DNA plasmid, but the protein format that is produced and secreted will make sure that this ends up in the antigen-presenting cells, both through the attraction and to the binding to the surface receptors of the antigen-presenting cells. And also this important feature that we have, that we ensure that it is a very effective cross-presentation to CD8 T cell responses. So it is a completely different mode of action, which is the reason why we see a much stronger immune response.
Is key to this technology, activating CD4 and, as you show, particularly CD8 T cell. Will the Neoepitope selection for the Vaccibodies vaccine be the same as used for other vaccines?
Yeah, so this is back to the core point. I think it's we observe something new here, that a neoepitope is not a neoepitope. It really depends on the format that you deliver the neoepitope in. So, I don't think you can rely on the exact same prediction tool. I think this is due to the fact that you deliver a neoepitope to different cells, different antigen-presenting cells, and that the neoepitope is then processed and presented differently. And also the distribution on MHC class 1 versus MHC class 2 is of importance here to the immune responses. So if you have a technology that can process and present the neoepitopes on MHC class 2 to a much better than extent than on MHC class 1, you need to find a neoepitope prediction tool that reflects that part also in the selection process. However, as with the Vaccibody, where we observe that we can elicit a very strong CD8 T cell response to the neoepitopes, we can focus also on predicting the strong CD8 T cell epitopes. The future will show.
I'd like to once again thank Dr. Fredriksen for her presentation. I'd also like to thank Labroots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. Email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.